Picture a tiny, a docile virgin female mouse. If a strange mouse just, wanders into her territory, she might sniff it, maybe follow it around a bit out of curiosity.
Yeah, or just ignore it entirely. They naturally avoid conflict.
Right. But if you fast forward a few weeks, it's the exact same mouse, just one day after giving birth. That stranger wanders into the cage again. And this time, our once docile mouse instantly transforms into this fierce, fearless warrior.
Oh, she launches a rapid, just relentless attack to drive the intruder away.
It's a stunning behavioral metamorphosis, and it happens almost overnight. So ask yourself, how does a brain completely rewire itself to protect its young, and then, just as mysteriously, switch that aggression off when the babies are gone?
It is the absolute definition of biological switch flipping. And for a long time, neuroscience just treated this like a black box.
Like, we knew it happened, but not how.
Exactly. We knew the behavior changed, but the underlying mechanics were a total mystery. We're talking about a massive, rapid reorganization of neural circuitry here. People often find it comforting to think of the adult brain as a static, hardwired computer.
Right.
But observing this maternal phenomenon shatters that illusion completely.
Welcome to today's deep dive. We are unpacking an incredibly fascinating April 2026 paper from the journal Nature that finally, opens up that black box.
And they did it beautifully.
They really did. The researchers managed to map the exact neural circuitry and the chemical signals controlling this dramatic rise and fall of maternal aggression. We are going to look right under the hood of the brain, exploring every physical wire and chemical switch involved in this transformation.
Oh, what stands out immediately in this paper is that it isn't just a localized story about aggression. It's a detailed mapping of how the brain uses multiple forms of plasticity to adapt to the real-time life or death needs of an infant.
Yeah, it's a survival story.
It really is. We're going to break down the exact mechanisms behind nature's most fiercely protective instinct. And the methodology they use to prove it is incredibly elegant.
So looking at the anatomy they mapped out, I was struck by just how granular they got. They didn't just point to a general brain region like the posterior amygdala and, call it a day.
No, they zoomed way in.
They pinpointed a very specific cluster of cells, the PAESR1 cells. These act as the threat detector.
Right.
And these are the cells expressing estrogen receptor alpha in the posterior amygdala. They spot the danger and essentially give the command to attack.
But a command is completely useless without a soldier to execute it.
Right.
So the researchers traced the projection from those PAESR1 cells deep into the hypothalamus.
To the VMHvl, right?
Yes, specifically to the ventrolateral part of the ventromedial hypothalamus.
Yeah.
VMHvl, for short. And again, they isolated a very specific subpopulation of cells there.
The ones expressing neuropeptide Y receptor 2.
Exactly, the VMHv-LNP2-ER cells. That right there is our action center.
So we have our anatomical highway. Threat detector to action center, PA to VMHvl. And I find the approach they took to prove this highway actually controls the attack. It's just fascinating.
The dread in latest experiments.
Yeah. They use a genetic tool called dread-ins to chemically silence the projection from the PA to the VMHvl, basically putting a chemical lock on the signal. And the moment they turned that specific pathway off, the lactating mothers completely stopped attacking intruders. But, and I think this is the real triumph of that experiment, what really matters is what didn't happen.
They didn't turn into zombies.
Right. The mothers still showed social interest in the intruder, and they still perfectly cared for their pups.
That specificity is a massive leap forward. Because historically, in behavioral neuroscience, if you wanted to see what a brain region did, you might lesion it.
Like, essentially creating localized brain damage.
Yeah, exactly. You damage it and observe the deficits.
Yeah.
But if a mother stops attacking because you damaged her hypothalamus, you might have also damaged her ability to nurse, or her motor functions, or just basic social recognition.
It's too blunt of an instrument.
Way too blunt.
Yeah.
By using DREAD-Ds, which is a completely reversible chemical lock and key system, they demonstrated that the brain has parallel independent circuits. You can surgically delete the attack command while leaving the care command perfectly intact.
It's incredible, but, proving a circuit is necessary for a behavior is really just the first step.
Necessity is only half the battle. They also had to prove it was sufficient to cause the behavior.
And that's where the optogenetics came in.
Right. So they infected those exact same PA projection cells with light-sensitive proteins, CHR2. By implanting a tiny optic fiber, they could shine a blue light and literally force those specific neurons to fire on command.
And when they shined that light on the lactating mothers, it triggered instant time-locked attacks on the intruder.
It was definitive proof.
Yeah.
The anatomical pathway drives the attack. The light goes on, the neurons fire, the mouse attacks. Light goes off, attack stops.
Okay, wait, I need to push back on this logic for a second based on what they did next.
Go for it.
The researchers took virgin mice that had never been pregnant. They put the exact same light-sensitive proteins in them. They shined the exact same blue light on that exact same anatomical highway, and nothing happened. No attack. The VMHvl is a known aggression center. If they're literally shining a laser into the brain to forcefully activate those neurons, shouldn't any mouse attack, whether it's pregnant or not? Why would a virgin mouse just ignore that massive artificial stimulation?
That seeming contradiction is actually the pivotal finding of the entire paper. It tells us something profound about the brain's architecture.
How so?
Well, the highway from the PA to the VMHvl is physically present in the virgin brain. The anatomical roadmap exists, but it's effectively dormant. The signal just isn't getting through with enough power to trigger the downstream execution.
Okay, let's pause and lock this in for you, the listener. Time for our first mini recap.
Good idea.
First point, the PAE-SR1 cells act as the command center. Second point, the VMHvl and pyapoi cells act as the action center. And third, this anatomical pathway is essential for maternal aggression, but it sits totally dormant in virgins.
Exactly.
So ask yourself, what physical changes must happen in the brain during pregnancy to turn this dormant pathway into a high-speed aggression highway?
To answer that, the researchers had to look at how the brain undergoes a massive hardware upgrade during pregnancy and lactation. They used a technique called patch clamp recording.
That's where you take a microscopic glass pipette and attach it to an individual neuron, right?
Yeah, you literally attach it to a single VMHvl cell. This allows you to listen to the electrical chatter of that specific cell and measure the tiny electrical signals, excitatory postsynaptic currents, or EPSCs, that are coming in from the posterior amygdala.
And they found two massive structural changes.
Two huge upgrades.
Right. So the first is synaptic strength, the actual physical connectivity between the general and the soldier. In virgin mice, the connection is incredibly weak. I like to think of it as having a terrible dial-up internet connection.
That's a perfect analogy.
The patch clamp recording showed that when the PA sent a signal, only about 12% of the VMHvl target cells actually received it in any meaningful way.
In the virgin state, the command from the PA is basically barely a whisper by the time it reaches the action center.
Yeah.
But in lactating mothers, that connectivity rate jumps from 12% to 79%.
Wow.
The synapses actually physically strengthen and multiply.
So the dial-up connection becomes high-speed broadband. Almost 80% of the action center cells are now securely receiving the attack orders. But the upgrade doesn't stop at just the connection between the cells.
No, it doesn't.
The second massive change happens within the VMHvl target cells themselves. They undergo a significant increase in intrinsic excitability.
Think of it as a CPU upgrade for the cell itself. The researchers found that the resting membrane potential, the baseline electrical state of the cell, that doesn't fundamentally change.
Oh, okay.
But when triggered by an incoming signal, the cells in lactating mothers fire significantly more electrical spikes or action potentials than the virgin cells do.
So even if you hold the incoming signal constant, the lactating cell's internal machinery just reacts far more aggressively.
Exactly. The cells are primed, they're hyperreactive.
That is a staggering physical transformation to undergo in such a short period. And what I found amazing in the sources is the precision of this upgrade. The VMHvl isn't just one monolithic blob of aggression cells. Sitting right next to our NPI2R aggression cells are cells expressing a totally different marker sector.
Right, and those are responsible for mating behaviors.
So if the brain just globally turned up the volume on the entire VMHvl, you'd get a very confused, maladaptive animal.
Oh, completely. But the researchers proved that the PA selectively boosts its connection only to the aggression-related NP2R cells during motherhood. It completely ignores the mating-related Saccharine cells.
That's so smart.
In fact, those mating circuits are actually suppressed during lactation, which makes perfect sense from an evolutionary and energetic standpoint.
Yeah.
A nursing mother needs her focus entirely on protection and rearing, not on seeking out new mates.
It's a total reallocation of neural resources. I do have to ask, though, how does this female hardware upgrade compare to male mice? because male mice, particularly the dominant ones, are naturally highly aggressive without needing to go through any sort of reproductive cycle.
Connecting this to the bigger picture of sex differences in the brain is quite revealing. Male mice are essentially born with this broadband connection already wired.
Really?
Yeah. Even without fighting experience, about 80% of a male's VMHvl target cells receive robust excitatory input from the PA. Their hardware is permanently installed. Females, on the other hand, employ an on-demand model.
So they only build this robust network temporarily.
Right, during motherhood, when it is evolutionarily necessary for the survival of their genes.
It's fascinating to think about pregnancy as a literal hardware installation process. You're taking a weak dial-up connection, turning it into broadband, and upgrading the processor of the target cells specifically for aggression, while actively ignoring nearby mating circuits.
It's incredibly precise.
But, time for our second checkpoint, because this brings up a massive puzzle. Point 1. Pregnancy strengthens the physical connections from the PA to the VMHvl. Point 2. The VMHvl target cells become highly sensitive. Point 3. It's upgraded specifically for aggression.
Perfect summary.
So here's a puzzle for you, the listener. If her brain is suddenly hardwired with this super sensitive aggression highway, why isn't she just violently attacking everything all the time? What acts as the safety lock keeping her behavior in check?
That is the crucial evolutionary constraint. Because aggression is incredibly energetically costly and it's dangerous. The hardware is there, but it requires a dynamic, real-time control switch.
Right. The brain needs a way to license that aggression only when appropriate.
Exactly. And that switch turns out to be oxytocin.
Oxytocin, the famous love hormone, the cuddle chemical that promotes bonding and trust. The irony of the love hormone acting as the trigger for a violent, relentless attack is just incredible.
It sounds totally contradictory if we hold on to the pop science definition of oxytocin. But in the context of this specific neural circuit, oxytocin acts as the vital gas pedal for maternal aggression.
How does that work physically?
Well, the researchers looked back up the chain at our threat detector, the PAESF1 cells. They mapped their genetic makeup and discovered that these cells are absolutely covered in oxytocin receptors, or OXTRA.
And during lactation, the expression of those oxytocin receptors significantly increases. The threat detector actively builds more docking stations for the hormone.
Yes. And when oxytocin actually binds to those receptors on the PA cells, it alters their physiology in a very specific way. It increases their input resistance.
Okay, if the synaptic upgrade we talked about earlier is the high-speed broadband connection, the increase in input resistance caused by oxytocin acts like a volume knob or an amplifier.
That's a great way to put it.
By increasing input resistance, oxytocin essentially ensures that any tiny threat signal the cell receives from the environment is amplified into a massive, unavoidable output signal. It makes the whole threat detection system wildly responsive.
That is a highly accurate way to visualize the biophysics of it. And to prove this wasn't just a circumstantial correlation, the researchers deployed both pharmacological and genetic interventions. First, they took a drug called L-371,257.
Catchy name.
Rolls right off the tongue. It's a potent oxytocin receptor antagonist. It physically blocks the receptors from working. So they injected this blocker directly into the posterior amygdala of lactating mothers.
And the maternal aggression dropped drastically. The latency to attack, the time it took her to decide to go after the intruder got much longer, and the duration of the attack plummeted.
But drugs can be transient, and they sometimes affect neighboring areas. So they went a step further and used CRISPR-like genetic tools to permanently delete the OXTRA gene exclusively in the PA cells.
And the result was identical, right?
Yeah. A massive, sustained reduction in aggression. But here's the critical control experiment they ran. They tried blocking oxytocin receptors downstream in the VMHvl action center. When they did that, absolutely nothing happened. Maternal aggression stayed high.
Oh, wow. That isolated the mechanism beautifully. It proves the PA, the threat detector, is the exclusive oxytocin control room for this behavior.
Exactly.
But wait, let's think about the real-world trigger for all of this.
Yeah.
Oxytocin isn't just floating around randomly. In mothers, it's famously released in massive pulses during nursing. The physical act of the pup's suckling is what provides the continuous supply of that chemical.
Right.
So what happens to this highly primed, heavily armed circuit if the pups are just removed?
That is the exact logical leap the researchers pursued next. They performed a pup separation experiment. They took lactating mothers, verified they were highly aggressive toward intruders, and then simply removed their pups from the cage.
And the results were pretty stark.
Very. Within 24 hours, the mother's blood oxytocin levels completely plummeted. And moving in perfect parallel with those plunging oxytocin levels, her aggression just vanished.
So when an intruder was introduced, she completely ignored it, reverting back to a behavioral state resembling a virgin mouse.
Yeah, but the critical twist, the detail that ties this entire neurobiological story together, is what happened to the hardware.
Oh, right.
The synaptic upgrades we spent so much time discussing, they didn't disappear.
The synapses were still strong, the dial-up hadn't returned, the broadband connection to the VMHvl was fully intact, the target cells were still intrinsically excitable, and the extra oxytocin docking stations were still sitting there on the PA cells.
Yep.
Basically, the sports car is fully functional, the engine is tuned, and it's sitting right there in the driveway. It just ran out of gas.
Exactly. The structural memory of the aggression circuit is maintained independently of the immediate behavior. To prove this unequivocally, the researchers performed what we call a rescue experiment.
The pups were still missing, right?
Right, pups are gone. The mother was non-aggressive. But the researchers used optogenetics again to target a completely different area of the brain. The paraventricular nucleus, specifically its oxytocin-producing cells, the PVNOX cells.
The paraventricular nucleus is essentially the brain's central oxytocin factory.
So with the pups gone, they shined a laser to artificially force that factory to pump out oxytocin into the brain, flooding those PA receptors.
And almost instantly, maternal aggression returned to full force, even without the pups there to physically stimulate nursing, providing the chemical gas was enough to rev the sports car back up and trigger a fierce attack.
Pure chemical tether. It is a brilliantly elegant need-based control mechanism. The physical presence of the vulnerable infant, mediated through the act of suckling, provides a continuous, dynamic chemical signal.
Which licenses the brain to act.
Exactly. That signal licenses the mother's brain to deploy this extreme, energetically costly behavior. When the infant isn't there, the chemical signal fades, and the behavior is safely locked away. Yet the architectural potential for that protection remains fully intact, ready to be deployed the moment the pups return and resume nursing.
Okay, final checkpoint to bring this all together for you. Point 1. Oxytocin acts as an amplifier on the PA cells. Point 2. Suckling pups provide a constant supply of this oxytocin. And point 3. Removing the pups removes the oxytocin gas, instantly pausing aggression without dismantling the hardware.
Spot on.
So think about the elegance of this. How does this real-time chemical tether ensure the mother only takes risks when her babies actually need her?
If we synthesize this entire journey, we're really looking at a masterclass in neuroregulation. We've seen how pregnancy hormones fundamentally remodel the brain's circuitry. You know, the what? Strengthening synapses, boosting cellular excitability from the PA to the VMHvl.
The structural foundation.
Right. But infant-driven oxytocin provides real-time, dynamic control over that circuit, the why and the when.
It prevents the mother from expending vital energy on aggression when the pups are safe in a nest elsewhere, or if, tragically, they're lost.
The brain only writes the check for aggression if the pups are physically present to cash it.
It makes you marvel at how beautifully efficient nature is. I mean, the literal physical act of nursing changes the chemical state of the mother's brain moment by moment, transforming her from a docile creature into a protector.
And back again.
But exploring this deep neuroplasticity leaves me with one lingering, provocative thought that the paper hints at, but sort of leaves open. If this aggression circuit is kept in this prime state, just waiting for the oxytocin gas to arrive, what happens when the pups grow up?
Yeah.
How long does that architectural memory, that broadband connection last after weaning is completely finished? Does a mother's brain ever fully revert back to that original docile virgin state, or does she carry the structural ghosts of maternal protection with her forever?
That is the absolute frontier of this research. We know the hardware stays intact for at least a day after separation, but mapping the long-term, perhaps permanent, legacy of motherhood on the brain's physical structure, that's a profoundly complex and fascinating question that neuroscience is really just beginning to untangle.
Something for you to ponder long after this deep dive is over. We started with a docile mouse transforming into a warrior. Now we know the exact wires and chemicals that make that magic happen. Thanks for taking this journey under the hood with us.